In this video, we'll discuss about the adaptive immune response involving humoral immune response and cell-mediated immune response. But first, let us look at the development of the immune cells involved in the adaptive immune response. The immune cells are B lymphocyte and T lymphocyte. Both of the lymphocytes were produced inside the bone marrow from stem cells. Bone marrow are spongy tissues found inside some of our bones, for example the femur. It is the stem cell found inside the bone marrow that will develop into lymphocyte. The lymphocyte that remain inside the bone marrow will mature into B lymphocyte or B cell, while the lymphocyte that migrates to and matures inside the thymus will become the T lymphocyte or T cell. Thymus is one of the lympho organ of the lymphatic system that secretes hormone thymosin, which is important for maturation of lymphocyte into T lymphocyte. Upon maturation, both B cell and T cell will migrate into lymphoid tissue such as the lymph node, spleen, blood, and lymphatic fluid. All the mature B cell and T cell have cell surface receptor called the B cell receptor or the T cell receptor. Each of these receptors are unique for each lymphocyte, and it can only bind to a specific antigen. Means that there are different types of B cell and T cell depending on the types of receptor that it possesses. Here we can see the general structure for the B cell receptor and the T cell receptor. Notice that both of the receptor is made up of variable region and constant region. The variable region also contain antigen binding site which will bind to antigen, similar to the structure of antibody. However, unlike antibody, the B cell and T cell receptor have an extra region called the transmembrane region. This is the part of the receptor that anchors or attaches the receptor to the cell membrane of B cell and T cell. Another thing that you'll notice is the B cell receptor is very similar in structure to the antibody compared to the T cell receptor. The main difference between B cell receptor and the antibody is the transmembrane region. This is why the B cell receptor is found attached on the cell membrane, but the antibody is secreted out of the cell. As mentioned earlier, the mature B cell and T cell will circulate throughout the blood and the lymphatic fluid and are concentrated inside the spleen, lymph node, and other lymphatic tissues. Mature B cell can become activated and differentiate into two different types, either plasma cell or memory B cell. On the other hand, the mature T cell, there are two types, either helper T cell or cytotoxic T cell. Both of the different types of T cell are differentiated based on their co-receptor, either CD4 for the helper T cell or the CD8 for the cytotoxic T cell. For the different types of activated B cell, the plasma cell are the one that secretes antibody, while the memory B cell are the one that have B cell receptor. Next, we'll discuss the process of activation of the adaptive immune response. Essentially, the process of activation involves activation of the lymphocyte. Only after activation, lymphocyte can become involved in the immune response. The activation process starts with a group of cells called the antigen-presenting cell, such as macrophages, B cell, and dendritic cell. Take up antigen, process it, and present the antigen fragments or the epitope on the cell surface. APC can take up antigen into their cytoplasm either by phagocytosis or endocytosis. Either way, the antigen will be taken into the cytoplasm where it will undergo processing and broken down into smaller pieces called antigen fragments. After that, the antigen fragments or the epitope will be presented on the cell surface of the APC by a group of protein called Major Histocompatibility Complex Class II or MHC Class II. MHC2 protein is unique to antigen-presenting cell. Means that only these groups of cells has the ability to present antigen fragment on their cell surface using this type of protein. Next, the complex made up of MHC2 and the antigen fragment on the cell surface will be recognized by the helper T cell or Th cell via their T cell receptor and the CD4 co-receptor. As mentioned earlier, there are different types of helper T cell based on the antigen binding site of the T cell receptor. Only the T cell with T cell receptor that is able to recognize the antigen fragment currently presented on the MHC2 will bind to the antigen fragment. The binding then triggers the APC to secrete a group of chemical called interleukin-1 or IL-1. Simultaneously, the Th cell will secrete IL-2. Both antigen recognition or binding of the T cell receptor with the antigen fragment and co-stimulation by cytokines, which is the IL-1 and IL-2, will lead to activation of helper T cell. Once activated, the helper T cell will undergo proliferation and differentiation into many copies of activated Th cell. At this point, we have many copies of helper T cell with the T cell receptor that is able to recognize that particular antigen fragment presented by the antigen presenting cell. Large number of the helper T cell will secrete large number of IL-2. IL-2 then recruits even more Th cell to the site of infection as well as activates the humoral and cell-mediated immune response. Humoral immune response is a type of adaptive immune response involving B cell, while the cell-mediated immune response is the type of adaptive immune response involving the cytotoxic T cell. That's it for the activation of humoral and cell-mediated immune response.